Okay, so in this video we are going to discuss for chapter 5 cellular respiration on part B structured question. So for question number 1, figure 3 shows step in glycolysis. Okay, step in glycolysis. As you know, glycolysis, it occurs in the cytoplasm. So at the end, and the product at the end produced is molecule A. Okay, so glycolysis basically it occurs in the cytoplasm. So the answer for number 1 is cytoplasm. Okay, so glycolysis, it consists of 10 steps. Okay, so the reaction that you see here, okay, is the reaction that produces fructose 1,6 biphosphate is step 2. Okay, the production of glyceraldehyde or glyceraldehyde 3-phosphate or G3P is the product produced at step 5. Okay, step 5. So step 5 will produce basically 2 molecule of G3P that will enter into step 6 until 10. So step 6 until 10, every intermediate and product you have to times by 2. So at the end of glycolysis, which is at step 10, it produces molecule A, which is pyruvate. Okay, so alright. Number 2, question 2. What happens to molecule A in the presence of oxygen? Okay, so molecule A, you know that it is already it is pyruvate. Okay, so pyruvate is actually a 3-carbon compound. Glucose is a 6-carbon compound. Okay, so it means that at the end of reaction 10, it produces 2 molecule of pyruvate. Okay, to get back the 6-carbon atom. Okay, so what happens to molecule A in the presence of oxygen? So the question worth 2 marks. So for the first mark you can get is by mentioning that A is pyruvate. And then what happened to pyruvate? It undergoes link reaction, or you can also instead of writing link reaction, you can also write undergoes pyruvate oxidation. Okay, so then you get 2 marks. But for further explanation, you may also include in your explanation if the question worth more than 2 marks. Okay, so you could also mention that the process of link reaction involve removal of carboxyl group. Okay, carboxyl group as carbon dioxide. Okay, as carbon dioxide, or it undergoes the process of decarboxylation. Okay, so and then the remaining, okay, because during link reaction carbon dioxide will be released, so the remaining 2 carbon molecule produced will be acetate. Okay, acetate. So here you can mention the remaining two carbon compound will be oxidized. Or it releases electrons. Okay, it releases electrons, and these electrons is actually used, okay, to synthesize NADH molecule. Okay, so link reaction, apart from it produces carbon dioxide, it also produces NADH. Okay, so so the next point you can mention here is that NAD plus, okay, will receive the electrons to form NADH molecule. Okay, and lastly, for the last point is the end product, okay, of link reaction, which is it forms from acetate. Okay, acetate is a 2 carbon compound that will combine with CoA to produce acetyl-CoA. So at the end, it produces or form acetyl-CoA as a result of acetate combine or binding to CoA. Alright. So that is the whole explanation of what happened during link reaction. Okay, if you are required to explain further. So number 3, calculate the number of ATP produced by this process after a complete oxidation of one molecule of glucose. So if you look at this question, it asks you to calculate. So you have to show your calculation basically to show the amount of ATP produced by this process. Which process? You have to refer back to the main question here, which is during glycolysis. So as you know, glycolysis, okay, it produces a net of 2 ATP. Okay, a net of 2 ATP, right? And then another product it produces is 2 molecule of NADH. Okay, so these two NADH molecule will enter into the oxidative phosphorylation stage to produce more ATP. So basically, uh, it, how do you know the amount of ATP produced during oxidative phosphorylation? So you have to know that for one molecule of NADH, it produces 2.5 molecule of ATP. And another molecule, which is one molecule of FADH2, okay, it produces 1.5 molecule of ATP. Okay, so basically, okay, so 2 NADH, okay, times with 2.5, you get 5, okay, 5 molecule of ATP. Okay, but the synthesis of NADH during glycolysis, it occurs in the cytoplasm. Oxidative phosphorylation, as you know, it occurs in the mitochondria. Okay, so the NADH molecule that was produced in the cytoplasm cannot enter into the mitochondria, but instead the electron from the NADH molecule, okay, the electron from the NADH molecule will be passed, okay, to the electron shuttle that are found in the inner membrane of mitochondria. So basically, for example, here you have a mitochondria, and then this is the inner membrane, and then outside you have NADH. So the NADH will have to pass its electron to electron shuttle, and the electron shuttle will pass the electron to either NAD plus or FAD molecule inside the matrix of mitochondria. So it depends on the cell. So if we are looking at liver cell, so the electron will be passed to NAD plus to form NADH molecule again. Okay, but if the cell is brain cell, okay, the electron will be passed to FAD molecule to form FADH2. Okay, so there are two ways of showing the calculation. Okay, so for one point, okay, so you have to mention, okay, the net production of ATP during glycolysis is 2 ATP during glycolysis, okay, through substrate level phosphorylation. Okay, so if we are considering this is a this is a liver cell. So liver cell, in the liver cell, 2 NADH, okay, will be produced from the glycolysis stage. You have to times by 2.5, okay, you will get 5 ATP molecule, and then this 5 ATP molecule you have to add with the 2 ATP molecule produced during substrate level phosphorylation of glycolysis. So 5 ATP that was produced during oxidative phosphorylation, okay, right, you have to add with 2, then you will get 7 ATP molecule. Okay, if you another way of calculating it, if you are considering it is as brain cell, so brain, instead of NADH will be converted to FADH2. So 2 molecule of FADH2 you have to times by 1.5, then you get 3 ATP molecule. You will have to add with this 2 ATP molecule produced through substrate level phosphorylation during glycolysis, then the total number of ATP produced is 5 ATP. Okay, so either calculation will be accepted. Okay, so this is for 3 marks. So 3 marks means that okay, 1 mark, okay, for the net production of ATP through substrate level phosphorylation, and then you showing that the NADH molecule has to times by 2.5. Okay, this calculation, and then the total ATP molecule that you get. Okay, so 3 marks. So again, same goes to the brain. So this is 1 mark, 1 mark, and then 1 mark. Okay, so either calculation is accepted. Okay, so citric acid cycle produces ATP via substrate level phosphorylation. Okay, explain this reaction. So citric acid cycle, the third stage of cellular respiration. Okay, so you have to explain how the ATP molecule is produced, okay, via substrate level phosphorylation during citric acid cycle. Okay, so if you refer to your notes, okay, to the diagram that shows citric acid cycle, you will find that the production of ATP is it occurs at step 5 of citric acid cycle. Okay, so during this stage, okay, it involves removal of coenzyme A from succinyl-CoA. Okay, succinyl-CoA. So removal of coenzyme A from succinyl-CoA. Okay, or forms succinate. Okay, so after the CoA is being removed, then if you look at the diagram, you will see that inorganic phosphate will enter into the reaction. Okay, so inorganic phosphate or phosphate group will displace, okay, displace CoA, okay, from succinyl CoA. Okay, inorganic phosphate displaces CoA from succinyl CoA. And then what is the function of inorganic phosphate that enters into the reaction? It is to attach to a molecule. We call it as GDP. Okay, so GDP will receive the inorganic phosphate to form GTP, and this GTP molecule will give one of its phosphate. Okay, GTP will remove its phosphate or transfer its phosphate, transfer the phosphate group to ADP to form ATP molecule. So basically these are the event that occurs during step 5 of citric acid cycle. Okay, so I repeat, CoA will be removed from succinyl CoA, and then the next item that enters is inorganic phosphate. This places the CoA, and then the inorganic phosphate will be added to GDP to form GTP. GTP will pass the phosphate to ADP to form ATP. So that's why the process is through substrate level phosphorylation. The phosphate is being passed from one substrate to another substrate until it forms phosphate. Sorry, until it forms ATP molecule. Okay. So next question. Alright. So question C, figure 4 shows an electron transport chain. Okay, an electron transport chain. So here you have complex 1, complex 2, complex 3, complex 4, and B. Okay, A is also it also function as an enzyme. Okay, as you can see from this diagram here, it produces it oxidizes. Okay, it oxidizes NADH. Okay, so name the enzyme A and B. So as you can see, the enzyme oxidizes NADH to form NAD plus. Okay, oxidizes means that the NADH molecule will release its hydrogen ion and also electrons. So the electron will be passed from one carrier to another carrier to another carrier to another carrier to another carrier until it is received by oxygen and the oxygen forms water by combining with hydrogen ions. Okay. So the enzyme that involved in the reaction of converting NADH to NAD plus will be NADH dehydrogenase. Okay, dehydrogenase. Right. Dehydrogenase meaning that hydrogen ion is removed from the NADH molecule. So enzyme B is basically, as you can see, it involves in the synthesis of ATP. So therefore B is ATP synthase. Okay, so what is the significance of electron transport chain for a cell? Okay, so the electron from the NADH or FADH2, okay, it has high energy. So this electron, as it travels from one carrier to another carrier, it loses energy, and so this energy is used to pump hydrogen ion, okay, from the matrix into the intermembrane space. So this region here is actually on this side here is the matrix of mitochondria. On this side region here is the intermembrane space. So matrix, you know already that it has low concentration of hydrogen ion. Intermembrane space, it has high concentration of hydrogen ion. So the energy from the electron will pump hydrogen ion from matrix into intermembrane space to accumulate the hydrogen ion in the intermembrane space to create proton motive force. Okay, so this proton motive force allows, okay, hydrogen ion to diffuse back from the intermembrane space back into the matrix. Okay, so the proton motive force will be used by the ATP synthase. It is a form of energy that the ATP synthase used to synthesize ATP from ADP and also inorganic phosphate. So what is the significance? So when a question asks for significance, in another word, it means that give the importance, okay, of the subject being asked. In this case, is the ETC, okay, electron transport chain. So what is the importance or significance of ETC? Okay. Number one, I'm just going to give the answer because the space is really small for me to write, so you have to listen carefully for the first answer. Okay, the importance of ETC is one, to synthesize ATP via oxidative phosphorylation. Okay, to synthesize ATP via oxidative phosphorylation. Okay, so the question is 1 mark, so that is enough for you for your answer. But you can but for your for more understanding, okay, on the importance of ETC uh, is that the ETC involves the transfer of electrons from one electron carrier to another electron carrier and causes energy to be released. Okay, for the second point. Okay, so I repeat, the transfer of electrons from one carrier to another carrier causes energy to be released. And then for the next point that you can add, the energy released, okay, is used to pump hydrogen ion from the matrix into the intermembrane space to create or to generate proton motive force. Okay, which is important during chemiosmosis to synthesize ATP. And then lastly, okay, another importance of ETC is that it is to regenerate electron carrier or NAD plus or FAD. So if you look at this diagram here, okay, so as NADH releases electrons, it regenerates the NAD plus or FAD, okay, FAD, so that it both can be recycled to synthesize or to create more NADH or FAD during during link reaction or during citric acid cycle. Okay. Number 3, describe how electrons from NADH is used to reduce oxygen during cellular respiration. Okay, so reduce means the oxygen molecule will accept electrons and also hydrogen ion. Okay, electrons and also hydrogen ion. So describe how electrons from NADH is used to reduce oxygen in during cellular respiration. Okay, so NADH and also FADH, FADH2 will undergo oxidation to release electrons. Okay, I repeat, NADH, in this case is NADH, or FADH will undergo oxidation to release electrons. Okay, so the electrons, for second point, the electrons will pass from one carrier to another carrier along the ETC. Okay, the electron will be passed from one carrier to another carrier along ETC, and then lastly the electron will be accepted by oxygen molecule as the last electron acceptor. Okay, so the oxygen then will combine with hydrogen ion to form water molecule. Okay, so you have to be careful in answering this question. The question asks you to describe how, so you have to describe the process. Okay, if you just write the equation, for example, 2 hydrogen ions plus half of oxygen molecule, you get a water molecule, then your answer would be incorrect because you just writing the equation. You are not explaining. Okay, so you have to explain because the question asks you to explain, describe how. Okay, so question number 2, what is the final electron acceptor in electron transport chain of cellular respiration? So we already looked in the previous question here, so the answer will be oxygen molecule. Okay, it functions as the final electron acceptor of electron transport chain before it produces water molecule as byproduct of cellular respiration. Cellular respiration occurs in a few stages. Okay, complete the table below by ticking a tick, okay, to the box to indicate the correct match of stages and products. Okay. So stages of cellular respiration is glycolysis, and then pyruvate oxidation, and then citric acid cycle, and then okay, you already know that the fourth stage is oxidative phosphorylation. Okay, so you have to determine what are the product of glycolysis, pyruvate oxidation, and citric acid cycle by placing a tick, okay, on the products. Okay, so glycolysis does not produce any carbon dioxide, does not produce any FADH2, so just leave it blank. And then pyruvate oxidation, it produces carbon dioxide but does not produces any FADH2. Citric acid cycle produces carbon dioxide, FADH2 is also produced. Okay, so each correct row worth 1 mark. Okay, so question C, figure 5 shows the citric acid cycle that occurs in animal cell. Alright, in figure 5, circle either K, L, K, L, M or N where ATP is produced by substrate level phosphorylation. Okay, so basically for citric acid cycle, you have to remember ATP, which was produced during substrate level, which is produced via substrate level phosphorylation during Krebs cycle occurs at step 5. Okay, so let's count the step. Okay, so step 1 is here, acetyl CoA that enters into the citric acid cycle to form citrate. And then step 2 is K, step 3, step 4, step 5, step 6, step 7, okay, and then step 8 actually, okay, 8. This diagram, please, it is actually a bit incorrect where the oxaloacetate will combine with acetate actually, so remove this one here. Okay, alright, I'm sorry, it is actually correct. Just okay, let's redo this labeling actually. Okay, alright, so basically during citric acid cycle, okay, the arrow should be, okay, in this diagram, acetyl CoA will enter here. Okay, it enters here, not here. Okay, so it means that from okay, so it means that from here is step 1. Okay, step 1 where acetyl CoA enters into the cycle, combines with oxaloacetate to form citrate. Okay, so here is step 2. Okay, step 2, citrate to form isocitrate. K is basically isomerization, and then isocitrate to alpha ketoglutarate is step 3, where carbon dioxide will be removed, okay, and also production of NADH molecule. Alpha ketoglutarate will experience another removal carbon dioxide and production of NADH molecule. Okay, so so this is step 4. L is step 4. M is step 5. So step 5 is where ATP will be produced via via substrate level phosphorylation. Okay, substrate level phosphorylation. So the answer would be M. Okay, so citric acid cycle basically it has 10 steps. So this is step 6, step 7, okay, step 8, and back to step 1. So that is the correct order actually. Alright, so just a little mistake here. It is not supposed to be here, as acetyl CoA will enter here in between oxaloacetate and also citrate here. Okay, so oxaloacetate is a 4-carbon compound, malate is a 4-carbon compound, fumarate is a 4-carbon compound, succinate 4-carbon, succinate CoA is also 4-carbon. Alpha ketoglutarate is a 5-carbon, isocitrate is a 6-carbon compound. Same goes to citrate, is a 6-carbon compound. Acetyl CoA is a 2-carbon compound. Okay, so acetyl CoA 2-carbon combined with 4-carbon of oxaloacetate produces citrate is a 6-carbon compound. Okay, so macam mana daripada 6 jadi 4 sebab hilang 2 carbon ni as carbon dioxide. Okay, so production of ATP basically occurs at M. Okay, so calculate the number of ATP. Just before that, and just to take note, okay, the production of another NADH molecule. Okay, another production of NADH molecule will occur, if I'm not mistaken, at step 8, NADH. Okay, so it means that for one complete cycle of citric acid cycle, it produces 1 ATP, which is produced here, 1 ATP, 3 NADH, and another product which is important, which is the production of FADH. If you refer to your notes, the production of FADH will occur at step 6. Okay, step 6. Here. Okay, production of FADH2. So another important product is FADH2. So this is for one complete cycle that consume or uses 1 acetyl CoA. But for one molecule of glucose, the citric acid cycle will occur twice. Okay, so you have to times all this by 2 to get the number of products for one molecule of glucose. So for one molecule of glucose, the product of citric acid cycle is 2 ATP, 6 NADH, and 2 FADH2. Okay, so these are the products of citric acid cycle for one molecule of glucose. So these NADH and also FADH will be used during oxidative phosphorylation to produce more ATP. Okay, the production of ATP here is through substrate level phosphorylation. Okay, so calculate the number of ATP molecules produced in C1 from one glucose molecule. Okay, show your calculation. Okay, so for 2 marks, you have to show your calculation for 1 mark. For the second mark is the answer. Okay, so how to show your calculation? Okay, so 2 pyruvate. Okay, 2 pyruvate or 2 molecule of acetate. Okay, so because acetyl CoA. Okay, 2 molecule of pyruvate or 2 molecule of acetyl CoA will enter into the citric acid cycle, causing the citric acid cycle to occur twice. Okay, to occur twice. So you know that one cycle produces 1 ATP molecule. So therefore, 1 ATP you have to times by 2 cycle. So you get 2 molecule of ATP produced via substrate level phosphorylation that occurs during citric acid cycle. Okay, which is C1 here. Okay, which is the main back to the main question, which is citric acid cycle. Okay, so one of the function of citric acid cycle is to produce NADH. Explain how this molecule is produced. Okay, so one simple explanation to explain how NADH is produced. If you understand the process of producing NADH is initially it is in the form of NAD+. Okay, so NAD+ will accept electrons, okay, and also hydrogen ions, okay, to form NADH. So therefore NAD+ is reduced to NADH. Okay, so 2 marks. NAD plus accept electrons and also hydrogen ions to form NADH. So by accepting electrons and also a hydrogen ion, NAD plus is reduced, okay, to form NADH. And then for further notes, okay, basically the reaction is catalyzed by an enzyme, okay, an enzyme. So the reaction, this reaction is actually catalyzes or uses the enzyme dehydrogenase. So you can add another point here, uses NADH dehydrogenase, okay, dehydrogenase, or you can mention that the process involved is dehydrogenation, through the process of hydrogenation. So how this process is how this molecule is produced is through the process of dehydrogenation using the enzyme dehydrogenase, okay. Or another point that you could include is that it involves the oxidation of compounds, okay. So compounds that are oxidized, meaning that the compound is losing its electron and also hydrogen ion. So the electron and also hydrogen ion of that compound will be given to NAD plus, okay. So NAD plus means that it is reduced. So the compound will be oxidized, okay. Oxidation of compounds, okay, compounds which involve removal of hydrogen ion. Question 4. What will happen to NADH once it enters into electron transport chain? So 2 marks. NADH will be oxidized, okay, oxidized, or NADH releases electrons to the ETC. So 1 mark. Or okay, the first point tadi, okay, oxidation of NADH, or releases electrons, okay, releases electrons. So for second point, it forms back NAD plus, okay. And number 3, another point that you could add is that it also releases hydrogen ions, okay. Or the whole thing you could also write it as NADH releases, okay, releases or will be oxidized and to form NAD plus, releasing hydrogen ion and also electrons, okay. So now you once NAD plus is produced, so now it becomes back as electron acceptor, okay. It becomes once again an electron acceptor, okay, becomes once again as electron acceptor. Okay, so it only wants two marks, okay. So any of these points. Okay, so for question D, cyanide, just a minute. Okay, cyanide is a deadly poison, forms a complex with cytochrome and prevent the transfer of electron to the final electron acceptor in the electron transport chain. So how would this affect cellular respiration? So the question is worth 4 marks. So basically for question D, it is actually an applied question. Okay, so you have to understand the whole concept of electron transport chain. Okay, so as you know, electron transport chain, it consists of several electron carriers. Okay, so one of the carriers are cytochromes, and the function of this cytochrome is to accept and transfer electrons. Okay, it is oxidized and then reduced. it is reduced and then oxidized. It receives and then releases the electrons. Okay, so in this case, cyanide, it prevents, okay, the transfer of the electron because it bound to cytochrome. So what is this? How does this affect cellular respiration? So for 4 marks, you could mention that the electron transport chain will unable to function. Okay, ATC unable to function. Second, no electron is transferred. Okay, because it is now blocked. Okay. Third, no proton gradient or no proton motive force will be generated. Okay, so the basic purpose of transferring electron from one carrier to another carrier is to pump hydrogen ion from matrix into the intermembrane space so that you can create a high concentration of hydrogen ion in the intermembrane space to create proton motive force. If the electron cannot be transferred, so no proton motive force will be generated. Okay, so the next point that you can write here is that chemiosmosis cannot occur. So when chemiosmosis cannot occur, the next point, no ATP or no energy will be produced. Okay. So because electron will not be transferred, cannot be transferred, so therefore NADH or FADH2 will not be reduced. Okay, will not be reduced. And then lastly, another point that you can write there is that no water will be produced also. Okay, so that's for question number 2 that worth total up to 14 marks.